Hello, my dear students, and welcome to this wonderful session. And I'm your tutor for today, Trapti Agrawal. Would like to give you a warm welcome. So, students, today we are going to learn a very interesting topic from your curriculum that is going to be the cell cycle and cell division. It's a very interesting topic, and it would be very helpful in your further classes. Also, you will be gaining a lot of experience by gaining this chapter. So, let's move ahead. So, students, when we talk about the cell cycle, we see a lot of important events that are happening. So, first of all, it's important to know that what is a cell cycle, right? So, cell cycle is basically an ordered set of events that, as I've already disclosed, that it is a set of events we see which are responsible for the cell growth, for the division purpose basically. And in this, we see that the cell is getting divided into two daughter cells. That is the main scenario that we are seeing in the cell cycle. And it is having different phases that we are going to discuss and that are responsible for the cell to divide into the two daughter cells, right? So the two main phases that are comprising a cell cycle are interphase and mitotic phase. So just like whenever we have to give any exam, we are like preparing for the exam, right? Similarly, when the cell has to undergo the division, right, the cell has to prepare itself. So the preparatory phase or the preparation phase is the interphase. So basically, in the interphase, the cell is preparing or making itself compatible to undergo the division purpose. That is, in the mitotic phase, it is getting divided. So it is preparing itself in advance in the interphase. So basically, interphase is responsible for the cell to grow so that the cell can grow itself and the DNA can be replicated. So the DNA replication and the cell growth is happening in the interphase, right? And in the mitotic phase, the cell is dividing to give two daughter cells. So that is about the overview of the cell cycle. Let's move ahead, students, and let's get to know that what are the different phases that are comprising this cell cycle, and let's understand about them. So here's a diagrammatic snapshot of the cell cycle. Let's understand about it. So when we talk about the cell cycle, students, we see different phases. Number one phase is G1 phase. It's a very important phase, students. That is the gap phase. It is also known as gap phase 1, right? And it is the longest phase we see. It is longest phase, students. And this G1 phase is longest because the maximum preparation or everything is going on in the G1 phase. Beyond in this G1 phase, as you can see that there is a G naught phase or the G0 phase, students. This G naught phase is basically the resting phase. It is the resting phase that we are considering in it. Right. This is the G naught phase. I will discuss about it in my upcoming slides. And there is S phase. S phase. In this S phase, we see that the DNA replication is happening. So first is the G1 phase. In G1 phase, we see a G naught phase in some cells that I will discuss. And then the second phase is S phase. In S phase, we see that the DNA is replicating basically. The DNA replication is taking place in the S phase. And the second one and the third one is the G2 phase, students. So this is about the interphase. Interphase is having three phases: G1 phase, S phase, and the G2 phase. And this G2 phase is the preparatory phase for the mitosis. After this interphase, the cell would be entering in the mitotic phase, where it is dividing into two daughter cells. Right. And this M phase is the mitotic phase, students. In this, we see the four important phases, that is the prophase, metaphase, anaphase, telophase, and the last one is cytokinesis. We will discuss one by one about them. So this was the small snapshot to give you an idea that how the cell cycle is, and how many phases are there that are comprising this cell cycle. Let's move ahead, students. So first of all, we will be discussing about the G1 phase. So when we talk about the G1 phase, students, we see it's a first gap phase, right? In this, we see that the cell is growing in size. The cell growth is happening basically. The cell is synthesizing a lot of proteins, right? And as I've discussed that it's the longest phase, it happens for 11 hours. Basically, 11 hours it is happening for, right? And it is producing new organelles. Basically, the new organelles are developing in the G1 phase, right? And we see that it is performing a lot of functions like the protein synthesis, energy production, or it is preparing the cell to undergo the further, further processes. Like DNA replication is not taking place in the G1 phase, but the preparation of the cell is happening in the G1 phase, so that the DNA can be replicated in the S phase, right? Cellular content, like including the chromosomes, everything is getting duplicated. That is all happening in the G1 phase. So in G1 phase, we see the cell is metabolically active. A lot of metabolic activities are happening inside the cell, and it is continuously growing. But there is no DNA replication that is happening in the G1 phase. DNA replication is happening in the S phase that we have already discussed in the snapshot, right? So that is about the G1 phase. Let's move ahead, students, and let's get to know about the next phase that is also very, very important. That is the G naught phase. G naught phase is also important, students. Why it is important, I will discuss. G naught phase is also called as the quiescent phase, and it is also called as the resting phase. We call it as a resting phase. Why? Let's understand. So there are some cell, students, which are not dividing, right? And these are not exiting the G1 phase, and these are entering into an inactive stage, into a resting phase. When the cell is actually resting, that phase or that stage is called as the quiescent stage. Although the cell is metabolically active, it is active itself, but it does not proliferate. It is not proliferating further. That's why it is stuck, or you can say it is in a quiescent phase. So that is about the G naught phase that we see in heart cells and a lot of cells other, right? So that is about this G naught phase and or the quiescent phase. Let's move ahead, students, and let's understand about one of the important phases of the cell cycle, that is the S phase, or we can say it as a synthesis phase. So in this S phase, the most important point is the DNA is getting replicated. The DNA replication is taking place in the S phase, right? And it is ensuring, or it is making sure that the new cell is having the same, or you can say identical genetic material. That's how it is happening. That's why the DNA is getting replicated. The same replica, or you can say the same copy of the DNA is forming in the S phase. So that when the two daughter cells are forming, they are making sure that the same DNA is transferring into the two daughter cells. That's how it is happening. And the key enzymes or proteins that are responsible for the DNA replication, these are all present in the S phase, right? And in the S phase, one more important thing is happening, student. The cell is checking, or the cell is making sure that the DNA is not having any error, because it's important. Because when the cell division is taking place, it's important to check whether whether there is any error or any damage is not there, right? Any dimer formation or something like not there, so that cell can smoothly work, right? And it is, and there are the repair mechanisms that are also responsible to make sure to that the damage is actually rectified, or it is completely relieved of. So that's how it's this S phase is working, students. And let's now move ahead towards the next part, that is the G2 phase. So in this G2 phase, students, we see a lot that is also called as the second phase, second gap phase, students. So in this second gap phase, we see that the cell is continuing to grow. It is producing the proteins that are necessary for the mitosis, right? And in this also, there is a checkpoint to make sure that all the DNA is replicated and there is no damage in it, right? So these checkpoints are again to make sure that there is no damage, or there is no lesion or something like that in the DNA, and everything is smooth going, and then it is responsible to make it ahead. So that's how this G2 phase is happening. And let's move ahead towards the next part, students, that is the M phase. So when we talk about the M phase or the mitotic phase, it is also one of the very important point that I'm going to discuss in the cell cycle, students. This mitotic phase is the most significant phase of the cell cycle. And here we see that the number of chromosomes in the parent and the progeny are the same. Like if the parent is in a diploid condition, suppose a parent is there, this parent is in diploid condition, right? So both the daughter cells, daughter 1 and daughter 2, these both are having 2n. The number of chromosomes remain the same. That's why it is called as the equational division. It is also called as the equational division, right? And in this, students, what is happening? There are two divisions that are happening. Karyo word is actually representing the nucleus. What it is representing? It is representing the nucleus. Whenever karyo word is coming, it is representing nucleus. So basically, the cell nucleus is dividing into the two identical nuclei. That is called as karyokinesis. And when we talk about the cytokinesis, cyto means cytoplasm, students. So here the cytoplasmic division is taking place in the cytokinesis, resulting into two daughter cells. The two daughter cells are forming. So that is about the M phase, students. And let's move ahead and let's understand about the next part. That is the important part. That is the cell division. So as I have discussed, students, about the mitosis, that mitosis is also called as the equational phase. Another cell division is also happening, which is happening in the germ cells. In the germ cells or in the gamete formation, students, or in gamete formation, the division that is taking place, students, that is the meiosis. What we call it as? We call it as meiosis, students, right? So meiosis is also called as the reductional phase. Why it is called as the reductional phase? Because, students, here we see, suppose that if the parent is having is in the diploid condition, right? So here in mitosis, we see that both the daughter cells are having the same number of chromosomes. We see the same number of chromosomes in the parent and in the daughter cell. But when we talk about the meiosis, students, or the germ cells, or we call it as the gamete formation, here we see the number of chromosomes are reducing, right? As I've discussed, suppose the parent is in a diploid condition. So the daughter cells are having haploid chromosomes. The daughter cells are what? These are having haploid chromosomes. So that is what is happening in case of meiosis, students. The number of chromosomes as compared to the parent, these are getting reduced. That's why it is called as the reductional phase, right? And in this, the two phases are there, meiosis 1 and meiosis 2. Right. So here we see the prophase 1, metaphase 1, anaphase 1, telophase 1, and the prophase 2, metaphase 2, anaphase 2, and the telophase 2. So that is about the mitosis and the meiosis, students. And let's move ahead and have a clear picture about the mitosis. So I have got a very small and a very concise diagram for you so that you can easily understand about it. Right. So I have mentioned all the important points for you. So here we see the prophase, metaphase, anaphase, and the telophase. So when we talk about the prophase, students, here we see that the chromosomes are what? These are getting condensed, right? And as you can see that the nuclear membrane is getting disappeared. That is the main or the characteristic feature that we see in the prophase, students. The spindle fiber formation is happening. The spindle fibers are getting forming. The centrosomes are responsible for the spindle fiber formation that is happening here. And the chromosome is consisting of the two identical copies of the sister chromatids. Here we can see that the sister chromatids are having these identical sister chromatids are intact in the chromosomes, right? And after the prophase, students, the next phase which is coming is the metaphase. So in this metaphase, we see that the chromosomes are aligned in a plate. As you can see, these are like aligned in such a plate. That is the metaphase plate. What we call it as, students, as the metaphase plate. And here we see that the spindle fibers are getting attached. What is this? This is the centromere, students. The centromere is responsible to adjoin these sister chromatids, and these spindle fibers are getting attached to it. Basically, to the centromere and the kinetochore. Kinetochore is also a structure which is there here in beyond the centromere, students. So these are getting attached with the spindle fiber, right? And after that, we see, students, the anaphase stage. In this anaphase, as you can see, that the centromere is getting break. The centromere is like this in this chromosome, right? It is getting break. So it is like the sister chromatids are like this only, right? They are getting separated. So here you can see that these are getting separated like this. And these are moving towards the opposite poles. You can see that these are the opposite poles, and here these are moving towards the opposite poles. And that how the cell is getting elongated, it is because of the spindle fiber, because the spindle fibers are getting elongated, and that's how these are responsible to pull up these sister chromatids, these separated sister chromatids towards the opposite pole. So that is how it is happening in the telophase, in the anaphase. And next is the telophase. Here we see that the chromosomes are getting decondensed. These are again getting decondensed to chromatin. And we see that the nuclear membrane and the nucleolus is again appearing, students. These are reappearing, right? These are coming back. And then spindle fibers, students, these are becoming disassembled. These are disassembled because they have already done their work. They have made the cell divided into two daughter cells, and that's how it is happening in the prophase, metaphase, anaphase, and the telophase. And now the nuclear division has already taken place. Now it's time for the cytoplasmic division to take place, students. And let's move ahead to understand about the cytoplasmic division, that is the cytokinesis. So in this, we see that the cytoplasm is getting divided and forming the two distinct cells, students. And here we see that in the animal cells, a cleavage furrow is formed. The cleavage furrow is responsible for dividing for the division of the cytoplasm to form the two distinct daughter cells. And in the plant cell, we see cell plate is there. The cell plate is getting forming between the divided nuclei to separate the cell wall, to develop into the separating cell wall. Basically, for the separate cells, the separating cell wall is there. That is about the cytokinesis, students, and this was a small snapshot about the cell cycle and the cell division. Hope you like it, students, and keep learning and keep shining like this. And thank you very much for joining us. Thank you.